Study on treatment efficiency of rubber processing effluent using Pseudomonas Fluorescens. Abstract. Even though rubber processing industry plays a significant role in polluting the water bodies, they contributes to a major portion of income in Kerala. Hence it is necessary to reduce the pollution from rubber processing industry. The present study was conducted to develop an eco-friendly process for the treatment of rubber processing industry effluent using Pseudomonas Fluorescens. Rubber processing industry consumes a large quantity of water which is disposed as wastewater. Economical and bio-friendly approaches are needed to remediate wastewater from rubber processing industry. Hence, a study was conducted with the aim of treating and disposing of rubber processing industry effluent that is rich in BOD, COD, PO4, NH4 and solids. The bacterium used in this study is Pseudomonas fluorescens, which will survive in the rubber processing effluent. The efficiency of both the species in treating the effluent was studied with varying bacterial dosage and contact time. The treatment using Pseudomonas SP yielded BOD, COD, TDS, ammonia and phosphate removal of 71.21%, 73.31%, 74.31%, 72.16% and 65.14% respectively. Introduction. Among the natural resources, water is considered to be the precious resource, hence it is required to be saved and maintained in its quality as well as quantity. An adequate way of conserving water is by recycling the wastewater from industries, as they are the major contributors of water pollution. As the wastewater from industries consists of large number of pollutants, it becomes difficult to maintain the water quality standards. Each freshwater body has an individual pattern of physical and chemical characteristics which are determined largely by the climatic, geomorphologic, and geochemical conditions prevailing in the drainage basin and the underlying aquifer. Rubber processing industry. Rubber processing is considered to be one of the major sources of income to our country. Out of 500,000 hectares of rubber plantation in India, Kerala state alone has 75.3%. During the processing of rubber, water is used for washing, cleaning and dilution. As per the studies, amount of effluent generated from an average-sized rubber factory producing about 20 metric tons of rubber was estimated as 410,000 liters per day. The present studies reveals that the latex concentrate effluent contained more suspended solids, dissolved solids, total solids, TS, and volatile solids, VS, which resulted in substantial growth of microorganisms resulting in high biochemical oxygen demand, BOD, and chemical oxygen demand, COD. Studies show that the amount of solids plays a significant role in the salinity of effluent. High BOD and COD value of the rubber processing effluent indicate that the TS and the effluents are mainly organic, especially fine particles of rubber hydrocarbon, with high oxygen requirements for their oxidation. Theoretical background. Pseudomonas fluorescens is a common gram-negative, rod-shaped bacterium. It is an obligate aerobe, but certain strains are capable of using nitrate instead of oxygen as a final electron acceptor during cellular respiration. Optimal temperatures for growth of Pseudomonas fluorescens are 25 to 30 degrees Celsius. The efficiency of Pseudomonas fluorescens in the bioremediation of rubber processing effluent is already studied. In that study, Pseudomonas fluorescens is isolated from the rubber processing effluent itself, and is used for the treatment of effluent. This organism is capable of reducing the contamination in the rubber processing effluent by neutralizing the acidic pH and also decreasing BOD, COD, and TDS. Experimental procedure. Rubber processing effluent for analysis was collected from a rubber processing unit in Thrissur. Characteristics of rubber processing effluent was studied in the college laboratory and the results obtained are as follows. The experiments were conducted as batch process using digital orbital shaker. 300 ml BOD bottles were used, each filled with 100 ml rubber processing effluent and Pseudomonas fluorescens with varying contact time and varying doses. Sample along with bacterial suspension was kept in digital orbital shaker at a speed of 120 rpm. After every 24 hours, the sample was taken for analysis of the required parameters. The bacterial dosage was varied as 1 ml, 2 ml, 3 ml etc. And contact time was varied as 1 day, 2 day, and so on. Results and discussions. 100 ml rubber processing effluent was inoculated with varying dosages of Pseudomonas fluorescens was kept in a digital orbital shaker at a speed of 120 rpm and the samples were taken at regular intervals and the various parameters were studied. Effect of varying dosage of Pseudomonas fluorescens on effluent parameters. In this set of experiment the contact time was kept as 1 day and the revolution speed of the digital orbital shaker as 120 rpm. At a dosage of 7 ml pH got increased from 4.7 to 7.5 which is in the desirable range of drinking water, 6.5-8.5. A better BOD removal is obtained at a cell dosage of 7 ml with contact time as 1 day at a revolution speed as 120 rpm. Approximately 71% removal in BOD can be obtained using Pseudomonas fluorescens at a dosage of 7 ml. As the dosage of Pseudomonas fluorescens increase the removal of BOD also increases, but after a dosage of 5 ml the reduction in BOD becomes constant. This is due to the increased dead cell mass. As the dosage increases, the percentage removal in COD increases and after dosage of 6 ml the percentage removal moves to a constant value. Hence 7 ml is considered to be the optimum dosage required to attain a better COD removal which gives a BOD of 5025 mg/L. More than 73% removal in COD is obtained at 7 ml with 1 day as contact time and 120 as the revolution speed in digital orbital shaker operating at 300 C. The effect of dosage of Pseudomonas SP on various parameters were obtained as follows. Also the removal of TDS was also studied using various dosages of bacterial inoculum with a contact time of 1 day and revolution speed of digital orbital shaker as 120 rpm. As the bacterial dosage increases the TDS removal also increases. In the initial stage itself, better removal efficiency is obtained which meets the desirable limit of 500 to 1000 ppm. Initial value of TDS was 2240 mg/L which got reduced to 575.45 mg/L with an efficiency of 74%. At a bacterial dosage of 7 ml, 76% removal of ammonia is obtained i.e. from an initial value of 97 mg/L, it is reduced to 22.95 mg/L after 24 hours of incubation. An initial removal of 67% was obtained at 1 ml dosage, 2%, after which the ammonia removal increases as the dosage increases. Effect of varying contact time on effluent parameters. Contact time of the experiment was taken as another varying parameter. In this set of experiment the cell dosage and revolution speed of digital orbital shaker were kept constant and the parameters were analyzed after the experiment. The contact time was varied as 1 day, 2 day etc. Dosage of Pseudomonas fluorescens was kept as 7 ml with a constant revolution speed of 120 rpm. The above table shows the effect of varying contact time in various parameters such as BOD, COD, and TDS. From the table, it is clear that contact time has no significance in the case of BOD removal and also in the case of COD removal. That is, maximum removal of BOD and COD was obtained at a contact time of 24 HRS with dosage of 7 ml Pseudomonas fluorescens at a revolution speed of 120 rpm. As the COD and BOD removal and remains constant after 3 days, the treatment was stopped. The removal of ammonia and phosphate increases with increase in contact time. But after a period of 3 days the removal remains constant, hence a contact time of 24 HRS was considered optimum with 7 ml dosage and 120 RPM as the revolution speed. Conclusions. For the giver sample of rubber effluent, 7 ml dosage of Pseudomonas fluorescens gives better treatment efficiency. It is also observed that varying contact time doesn't show much effect on the treatment efficiency of Pseudomonas sp. Increase in dosage of bacterial sp. increases the removal efficiency but the removal rate remains constant after a particular dosage. Here it is 7 ml.